Hello, I'm Sarah Barth, Executive Director of Sempervirens Fund. Thank you for joining us in the most recent in our Under the Redwoods webinar series, in which we explore all things related to redwoods, their history, art, science, ecological role, you name it, the poetry of redwoods. We've covered it all. Thank you to Sharf Investments, who has been sponsoring this Under the Redwoods webinar series. We are really grateful for their support. Over the course of our 125-year anniversary, we've been looking back, and this speaker is going to help us look back into deep time. Dr. Zane Moore is a plant biology and genomics expert who specialized in redwoods. He is actually, you can see him in the Giants Rising screening. He's completed his degree at UC Davis, so he's Dr. Zane Moore, and he's currently a biology teacher in San Jose. One of the things he's probably most known for in the redwoods world is that he was instrumental in the first effort to sequence the redwood genome and to study mutations in redwoods and help us understand a bit about what they tell us about these trees, about their past, and to some degree about their future. So with that, I'm really excited to have Dr. Zane Moore join us. Welcome, Zane.
Thanks so much, Sarah. Thank you for the introduction, and thank you all for joining. I really appreciate the invitation. So I wanted to give a quick background of how I got interested in redwood trees. It was all in the Santa Cruz Mountains, so I figure a lot of us, for a lot of us, the Santa Cruz Mountains is near and dear to us. So I got interested in redwoods actually through my interest in trains. So the Forest of Nicene Marks has a lot of railroads that were used for logging. The Forest of Nicene Marks is in Aptos, and there are train tracks that crisscross that entire park, or there were back 100 years ago. And so learning about those and finding where the train tracks were and that sort of thing got me kind of started exploring in the Santa Cruz Mountains. And in doing so, I ended up finding that Nicene Marks was not completely clear-cut. There were a number of old growth trees that are left here and there scattered throughout the park, little remnants, little veterans. So I ended up looking for some of them, and one of them is my favorite redwood tree still to this day. It's this tree up by Sandpoint Overlook in Nicene Marks. It's off trail, it's gigantic. On the widest point on the ground, it's 20 feet diameter, but at breast height, it's probably something like 14, 15 feet. It's been burned out by fire. It's charred. It doesn't have a lot of merchantable lumber in there. And the top actually breaks about 20 feet above my head. And there's a big 6-foot diameter arm or reiteration coming out with 4 vertical trunks coming out of it. It's an amazing tree. But in doing this, I ended up getting really interested in finding like the weird, unique trees that were left and trying to figure out why these trees were left. At the same time, another weird thing that I was really interested in that I learned about through a KQED Quest program on YouTube was albino redwoods. And so this is the first albino redwood I ever saw. It's in Henry Cowell. Some of you all might be familiar with it. And so that's like a weird genetic anomaly. It's like needle in a haystack, kind of like these old growth trees in Nicene Marks were needles in haystacks of all the other, second growth forest in that park. And so I got really interested in all of this. It's like it opened it up this world of exploration. At the same time, in Nicene Marks, there's a group of trees called the Twisted Grove. That was another weird oddity. And people debated about whether that's genetic or whether that's Loma Prieta earthquake sliding and the trees responding. I think it's genetic. We don't know, but I think it's genetic. I didn't know at the time, but my trip to Camp Jones Gulch property was the first time I saw a stump over here on the right-hand side that was cut with twisted, wonky redwoods coming out of the stump, each one of the sprouts kind of weird and not growing straight. And this is the first time I thought, maybe the twisted grove might be its own genetic thing and not a Loma Prieta earthquake remnant. Okay, on the left-hand side, we have another weird genetic anomaly. These are fasciations, and we won't have time to talk in detail about fasciations. What they are, though, is they're inside the bud of any plant. Any plant can do this. Inside the bud of any plant, that bud is unable to branch. And so in trying to branch, it ends up sticking together and creating this ribbon of growth and this elongated stem that kind of looks like a coral. So anyway, back in high school, I'm like, what is this? This is cool. How bizarre. And um, we know why this exists. For those plant biologists out there, it's a mutation on the shoot meristemless gene and the clavata gene together that cause something like this. But we don't have time to talk about that. We won't get into that. We have more important things. So at the end of high school, I ended up meeting and working with Steve Singer, pictured here. He's a marbled murrelet ecologist and was on the science advisory board of Sempervirens Fund. And he has old growth maps of the entire Santa Cruz Mountain region. It turns out there's 11 square miles of old growth in the Santa Cruz Mountains, publicly and privately held, a vast majority of it in Big Basin. So I spent a summer waking up at 4 a.m. And going up every drainage in Big Basin, going up every conceivable place where there's old growth that I could publicly and legally access in the Santa Cruz Mountains. And I did this for a whole summer with a laser rangefinder you see here, looking for tall trees. Okay. I ended up finding through this effort the tallest trees in Big Basin. This is a 328-foot tall coast redwood. It's the tallest tree in the world from Big Basin south of any species, taller than any eucalyptus, taller than any tropical tree. It was burned in the fire, lost all of its branches, but there are green sprouts now at 331 feet tall. So it is still growing tall after the fire. So happy to report that. But back in high school, when I found this, no one believed it existed here. And so I worked with Michael Taylor, who's pictured here. He's the guy who discovered the world's tallest tree. And we measured it with lasers, or he could confirm my measurement with a laser, as it being 328 feet tall. And for the people who like metric, that's exactly 100 meters. And so that was very exciting. It was so exciting that Susan Blake, if any of you know her, she was the interpreter at Big Basin. I love this photo of her hanging out, totally content in finding this tree in her park, or the park that she interprets at. So really, really special. From there, I learned how to analyze LiDAR. Now, what LiDAR is, it's very expensive. You get it from a plane pummeling lasers down at the earth, about 10,000 laser beams a second. And those returns, where the laser reflects off of foliage or ground or anything else it hits, those returns get saved as a three-dimensional point cloud, as you see here on the left. On the right, we can take these three-dimensional point clouds and look down on them and color code the dots based on their heights. So here, black is where there's nothing growing there. It could be a meadow, it could be a creek, something like that. Red and white are very, very tall trees, over 270 feet tall. This is the tallest second growth forest in Nicene Marks, with the tallest second growth redwood tree being around 287 feet tall, making it one of the tallest second growth conifers. The park also has the tallest second growth conifer, period. It's not a coast redwood, it's a Douglas fir, 110 years old, 301 feet tall when we last measured it a couple years ago. So super amazing tree. It's getting closer and closer to the record for that entire species, which grows all the way up into Canada and all over the Western US, but it's in one of the drier parts of its range. Okay, so through this, I ended up finding the tree that I would work and study my PhD on, and the first tree that I want to introduce to you, the Redwood Genome tree. This is a tree that you'll see a person standing at the base. That person's my dad. It's 352.6 feet tall. And it's the southernmost 350-foot redwood, southernmost 350-foot tree, and tallest tree in the world, above like 1,000 feet in elevation. It breaks up here at the top into a bunch of different reiterated trunks. That branch up there at the top, or the limb coming off, is 4.5 feet diameter, 260 feet up, which is just amazing. Anyway, this tree ended up being the tree we selected for the Redwood Genome Project, which was a project funded by Save the Redwoods League through members at Johns Hopkins and at UC Davis. My sister also drew a pencil drawing of this tree that's accurate with all the measurements and the photos. So we have photos of this tree. Steve Sillett climbed it and measured the trunk with tape. And so this is an accurate representation of that tree. We know it's 1,392 years old at minimum. And we know this because Steve Sillett took a bunch of core samples from the trunk, 44, I think, in all, from bottom to top of the tree. And you can see there's a bunch of rings there. When we sequenced it, this is a shot of the film Giants Rising. We sequenced this middle nutritive tissue. The nutritive tissue is less genetically complex than the rest of the tree, and we'll get into why that is in just a second. And I ended up sequencing 14 different branches in the tree. These are the different branch locations. You can see that the tree has two tops, S and Q. Those two tops both independently reach 350 feet from that break at 260 feet lower down. So amazing tree, a phenomenal tree. Okay, so why sequence all these branches? Well, and one other thing before why sequence all the branches, we also looked, I sectioned buds from different heights in the tree to look and see how the cells divide and how many cell divisions occur in a redwood tree. That was a really tricky thing to do. And anyway, we aren't going to talk about that because that's not a genetic part of the talk, but that's okay. So why sequence all these branches in the tree? When I was an undergrad at Colorado State University, my professor invited me to go to his graduate level class in plant morphology, ecological plant morphology. And this is where I first heard of this concept of thinking about a tree as a population of branches. And I think this is an important concept for the rest of the talk. A tree is a population of branches that we can describe demographically, as in branches are born, right? There could be a branching event or a new baby bud and it's born. A branch grows and lives out its life. A branch can die, right? And so we can talk about these things demographically. So learning about this kind of from a physiological perspective, I thought it was interesting to also consider that when we think of albino redwoods, these are branches that have mutated and changed. And so maybe not only is a tree a population of branches, but are they genetically different branches? That's one of the reasons why I wanted to study this. The other reason that this matters is that branches reproduce. And so, and sometimes an entire tree can just be one branch. This is a branch of this is the largest known horizontal limb in the world of any tree species. It's in a land protected by Sempervirens Fund in the Santa Cruz Mountains. And there's a person there for scale. It's 9 feet diameter. Okay, so sometimes trees are branches, but what I'm trying to say is a tree is a population of branches, not just one branch. Okay, those branches can be genetically unique. And in this case, we have an albino redwood branch high up in a tree that's producing male cones. Albino redwood branches can also produce female cones. And the crazy thing about this whole thing is that those are reproductively viable and will create little baby seedlings. Here's a seedling here that's albino. And so what that tells us is that mutations in branches are heritable. If a branch changes, it is possible that that change, if it's genetic, can be passed on to its offspring, which I think is fascinating and poses a bunch of questions. So we got to talk, when we're talking about redwood genetics, I'm sure a lot of you have heard the term hexaploid, and so we need to talk about this. So you and I, humans, are diploid. We have 2 copies of every chromosome, 2 copies of every gene, with the exception of the X and Y chromosomes, where we might have 1 copy or 2 copies, depending on who you are. On the bottom here, we have, so this is a representative of a human body cell here. On the bottom, we have representing a redwood body cell. It's hexaploid. It has 6 copies of every chromosome, 6 copies of every gene. Okay, so let's talk about how a life cycle might happen. So let's look at the human body. Human body produces sperm and eggs through meiosis, and those sperm and eggs have 1 copy. They're haploid. And so if a mutation occurs in the germline of your own body, which, newsflash, 40 to 60 mutations do occur in your germline that you pass on to your offspring, that happens through this way. If a mutation occurs in the body, that means 50% of your gametes, sperm or egg, are going to have that mutation. Now, if that mutation is lethal to a sperm or egg or really damages or impairs its function, it can actually kill the sperm or the egg. By killing that sperm or egg, that mutation gets selected out of the gene pool. And so the next generation, all those sperms or eggs that have that mutation aren't going to be there. And only the ones that do not have the mutation go on to make the next generation. And so you can adequately filter out mutated chromosomes, mutated genes. You can adequately filter those out through this simple sexual reproductive process. That's regular sexual reproduction, what we all know and love. The second way, now we'll look at hexaploids and redwoods. So I told you about the seed of a redwood tree, and that we looked at a component of the seed because it was genetically simpler. We looked at a component of the seed called the megagametophyte that has 3 copies, which is this cell down below. Those 3 copies end up making sperm, if it's pollen, or in the megagametophyte's case, eggs. A redwood tree can have dozens of eggs in a single seed and can have multiple pollen grains that pollinate it. So you can have multiple embryos inside of a redwood seed. The theoretical maximum is kind of insane, somewhere in the 50 or more. But usually only 1 or 2 survive. All right, so this nutritive tissue here that we sequenced has 3 copies, and that's where all the eggs are. Anyway, if a mutation occurs in the redwood body in a branch, let's say, and then that branch goes on to make pollen or seeds, then maybe one of those copies, in 50% of them, ends up mutated. But notice, we have 2 other functional copies, and so that seed does not die. Selection can't happen. No skull and crossbones here, because as long as there's 1 functional copy, in most genes and in most scenarios, if there's 1 functional copy, that thing can go on to reproduce in the next generation. So let's imagine one of these sperms and one of these eggs with a mutated copy come together. Now we have 2 mutated copies. And some of those sperms and eggs from that next tree are going to have 2 mutated copies. And if those 2 come together, we can have 4 mutated copies. And then you can imagine now, finally, we might have some sperms or eggs that have 3 of these mutated copies that have finally die, where selection can act on them and get rid of them and remove them from the population. But you see what the situation is with the tree that produced this. The tree does not have a lot of functional copies. And this is what we call mutational meltdown, when mutations accumulate in a population and to the point of extreme, to the point where a lot of the copies in the population are this mutated copy that's harmful. This can happen typically in small populations. Okay, so the question is, doesn't this seem kind of bad for redwood trees? I guess they would probably not want to mutate very quickly, right? The next thing I want to mention is that this hexaploidy, the one other thing that's important is that it is the reason why redwood individuals can live so long from a genetic perspective. It's because when you have 6 copies of every gene, you need 6 different mutations on 6 different copies to wipe that out and end up killing the tree, right? That takes a long, long time. And especially if these are in separate branches, you might, if all 6 mutate in one branch, it might just kill a branch, not the whole tree. So this is how redwood trees can just live forever because they can just, whatever, they have a bunch of copies of everything. Okay, so my question, and we'll come back to this, is do we ever see a single copy like we see in human sperm and egg? Do we ever see a single copy exposed to selection in redwood? Theoretically, no, but we'll get back to this. Okay, so why don't redwood forests suffer from mutational meltdown? Why is not every seedling or so many of the seedlings that get produced in a redwood forest come out to be albino or have some other really gnarly mutational problem with them? Well, let's sequence them and find out how fast is the mutation rate of redwood. When I ask this question to audiences, like state your answer. Do you think redwoods mutate quickly or slowly? Most people would say slowly. It turns out redwoods mutate very quickly. We sequenced 0.2% of the genome and found in these 14 branches, and I found 14, in the 14 branches, and I found 30,000 mutations in those 14 branches in that 0.2% of the genome. You can do the math. That means that redwoods have millions of mutations in their crown. If we were to look at the whole genome, which is super expensive to do because it's so big, redwoods would have millions of mutations in there. Also, we only sequenced 14 branches. Think about all the other branches we didn't sequence. Branches are reservoirs of genetic diversity, and they're reservoirs of genetic diversity in a forest. If you have an old growth tree, think about how many branches there are in that old growth tree. There's a bunch, and they've been doing their branch thing for a long time. And so they've been accumulating mutations. They might have different reproductive success. Who knows? Do they have different reproductive success? Well, we looked at cones from the 2 tops, and it turns out, yes, in fact, they did. So we looked at S and Q here, the 2 tops, and we found that with one of the tops, we had smaller cones, we had lighter seeds, we had fewer living seeds with that living megagametophyte that makes the eggs, and fewer germinating seedlings, I should say, okay, versus the other top. And you notice that there's quite a big difference, especially when we look at how many seeds are living. And if we look at how many that germinated, they're also pretty different. And so, wondering why that is, when we look at the genetic sequences, we find that one of the tops was missing a copy of chromosome 3, aka it mutated away and there's no more chromosome 3 in one of the tops. And that top had poorer seedling growth, smaller seeds, smaller cones, less reproductive output. I thought that was a cool result. We can interpret that result in this way, right? Where maybe the reason why we have less reproductive output is on the right-hand side, notice that our sperm and egg, they're mostly viable, they're mostly good. But on the right-hand side, we have, with that missing chromosome, maybe that was the only functional copy. And so we have more sperms and eggs that are dead or dying, or that selection can act against. So that's one interpretation. But redwoods do something that's, I think, pretty amazing. And this happens over long scales of evolutionary time. They do this thing called diploidization. If you need a new 15-character password, diploidization might be a pretty good one to pick. Diploidization is what happens during meiosis, where instead of all 6 chromosomes grouping up together, chromosomes group up only in pairs, and or in this case 4 groups of 4 instead of 6. And so here we have hexavalence, tetravalence, and bivalence. The hexavalent chromosomes act like 6 identical copies of every single gene, or mostly identical. Okay. Tetravalence and bivalence are indicative of mutations accumulating on chromosomes where they begin to pair up separately. And it's not a group of 6, but it's sort of 3 groups of 2 that have different mutations that look more similar to each other than the other members of that group of 6. And so I have this diagrammed here as different colors for this hypothetical chromosome. Notice in this meiotic figure over here to the right-hand side, we see 1 hexavalent. The rest of these are tetravalents, and then there's a lot of bivalents. What this indicates is that the Redwoods chromosomes, a lot of them look more like this right-hand colorful figure versus the left-hand all uniform, truly hexaploid or pretty equal copies for all of the different, for that chromosome. Okay, so more of them are starting to specialize. They're starting to, we call this neo-functionalization. That means new function. They slightly mutate differently, and so they slightly get different functions. Now, if we think about a missing chromosome in this case, instead of it looking like this, it might look more like this if it's a diploidized chromosome. What does this mean? This means that maybe the loss of a chromosome means there's only 1 functional copy, this pink one. And so sperm and egg that get the pink copy survive. Sperm and egg that do not survive. So it could look more like this and more like the right-hand side and less like this left-hand side. Okay. All right. Let me check the time. Okay. So what I really want you to take away from this talk is that a tree is a population of branches, and those branches are constantly evolving. And what happens evolutionarily could get passed on, or what happens in the crown of the tree could get passed on to offspring. Okay. So genetically speaking, the branch sort of acts as the Darwinian individual, the individual evolving, the individual that might change and that might have reproductive success or failure based on those changes. So my next question was, do we see any instances of mating success when there is a missing chromosome? If a missing chromosome occurs, can we make new offspring from that is missing a chromosome? So do either of these scenarios happen? It turns out, yes, they do. And this is the instance where it happens. So this is all from a single seed that was missing a chromosome. Every single tree in this image came from a single seed that developed a long time ago, and has been growing out ever since into this gigantic fairy ring that you see here. This is the Wunderlich clone. It's the largest redwood clone by area, by like ground area that it covers. And here's an image showing all of the different trunks and the diameters of those trunks. They're all in blue here. We mapped this with the Friends of Wunderlich Park and 10 volunteers. It took 3 days to map all this out. It took forever. You can see that the scale of the squares is 10 meters. And this is as if we're looking down on the clone from above. You can see the north arrow here. Each of these blue dots is a tree. And each of these trees is growing to roughly 30 or 40 meters in height. The tallest one is 44 meters. And then the other thing I'll point out is look at all these arrows that are on the ground. These are trees that have fallen over and are growing alive horizontally on the ground. I'll talk to you more about that in just a second. Very bizarre. So I want to thank the Friends of Huddard and Wunderlich Parks for the help. This clone covers 1.5 acres. It consists of 280 trunks, and it's 1,700 tons. It's heavier than the General Sherman by a little bit. And there may be heavier clones out there, but we haven't sequenced them. This is where it's located. So if you go on Google and look up Bear Gulch Trail on Google Maps, you'll see in satellite view this ring. This is the first way that I saw this clone. I was looking on Google Earth and I'm looking for big clones. You can see the clones from satellite, or what looked to be clones. And so I saw this thing and I was like, man, I hope there's like a really nice phenotype that would set all these trees apart that I could easily tell, oh yeah, this is all one thing. Well, turns out that the phenotype of this clone is that none of the trunks grow straight. Every single trunk grows at an angle. They start out growing straight until they start producing secondary growth, that is wood and bark. Once they start producing wood and bark, then the trunks start to lean and do this wonky, they're crazy, and they start falling over and it looks like pickup sticks. It's really wild. Time spent under these trees is borrowed time. We measured one of them, and in less than an hour after we were at the base wrapping a tape around it, it fell over and would have crushed us. And on its way down, it actually hit a bay laurel tree and gave the bay scent. The bay scent you could smell for like a quarter mile in any direction. It was amazing, but also terrifying. So don't spend too much time under there. The trunks can't grow straight. The second thing that's kind of weird is that they have abnormal wood anatomy. They don't make proper compression wood, which is that wood that buttresses the underside of a trunk or a branch. They can't do that very well, or their compression wood's all messed up. And so this might be one of the reasons why they fall over. Since we measured it, about 10 trees have fallen over just in the last like 3 years. So they're constantly falling over. So just again, standing under there is borrowed time. The other weird thing about their wood anatomy is they have weird resin ducts in there. So very bizarre. Next, the trunks can grow horizontally on the ground for decades. Some of these trunks, you can kind of see one of them down here in the bottom of your screen. Some of them have been on the ground since the 1980s or 1990s. And they've just been growing on the ground ever since then. We can tell that because that's the last tree ring that they produce, and yet their foliage remains alive. We think that this could be a way that they can expand outward, because if they fall outside of the clone and root out there, the clone can expand further out than if it were just sprouting little by little over centuries. And lo and behold, it's missing a copy of chromosome 4. Every single tree we sequenced 9 of them. This is with Sasha Nikolaeva at UC Berkeley. We sequenced 9 of them, and all of them are missing this copy of chromosome 4. So I hypothesized that probably chromosome 4 and the genes on there have something to do with this leaning phenotype, and chromosome 4 is probably more diplotized than some of the other chromosomes. We also know if we use the mutation rate that we found in that one tree that you saw earlier, if we use that mutation rate to try and date how old this tree is based on the number of mutations it's accumulated, this tree is about 28,000 to 30,000 years old. Or that's when it was a seedling. And then the final thing that's really cool is that I collected some female cones from it this last year, and those female cones are reproductively viable. They produced 3 seedlings. 2 cones produced 3 viable seedlings. A 4th one was produced, but it died because it probably had it looked pretty weird. But 3 of them are viable, making this the oldest like mother organism that I'm aware of. So kind of cool. Okay, so I want to leave you with this. What's an individual redwood? Well, we often think about the individual as being the tree, right? We see a redwood tree, you've seen one, you've seen them all, or so some say, and that is an individual. However, I'd like to challenge that view. We have individuality as branches, and an albino redwood is a great example of that sort of individual characteristic in a branch. We also have individuality on the far end of the spectrum being these large clones or large fairy rings. And so individuality is a big spectrum. And that spectrum spans large amounts of time. So branches can live to be, 1,000 years old. Trees can live to be 2,500 years old. Clones, this one might be 30,000 years old. If anything, they live a lot longer than individual trunks or individual trees. So they span these big time scales. They also span spatial scales. So a branch, the longest one I'm aware of, is 35 meters in length. It's in Prairie Creek State Park up in Humboldt, and it grows down parallel to the trunk, 35 meters, 114 or something feet, down to get sunlight in by a creek bottom. Then the tallest tree we know of is 116 meters tall. It was just recently climbed by Steve Sillett and measured again. It's still growing, thank goodness. It is illegal to go visit that tree. Do not try. And then finally, with clones, they can be up to 6,300 square meters or cover that area on the ground. So you have these spectrum of individuality. Going one way, we have genetic independence, right? The branches are more genetically independent per se than maybe the tree is. The tree is more genetically independent than, let's say, the entire clone. But then we also have physiological independence going the other way. Most of these branches are going to live or die by whether that tree lives or dies, right? Most of the trees in a clone live or die whether the clone lives or dies. There are examples, and we can talk about this in the Q&A, with climate change. There are examples of clones that are dying over here on near 280 here in the Santa Cruz Mountains, where like one tree starts to go brown and then they all go brown. So they're physiologically connected and kind of act together as we go towards the right of the spectrum. The longevity of the unit obviously gets a lot longer as you go to the right, and then the size of the unit gets a lot bigger as you go to the right. Okay, so that's what's fascinated me about redwoods since I started, is sort of their individuality and these different levels of hierarchical individualism, I should say, or whatever. I don't know how you say that. And to me, that's been the most interesting thing. We have time for questions. And I also, I need to first give my acknowledgments. So my PhD work, the work on the really tall tree, these are all the people that help contribute to that from a bunch of different institutions. I really appreciate that. And then the Wunderlich clone, you can see the list of volunteers here in the upper left. Without these people, it would have taken me a month or maybe 2 months to measure it. It would have been impractical. And so I really, 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 really thank all these people for all the hard work they did to put in so that we could actually measure the clone. I also want to thank the collaborators who are helping me, Allison Carroll with the clone, Sasha Nikoleva, and Christian Cho. And you can see that a number of different universities have partially funded a bunch of different parts of this thing. So I want to thank everybody for that. And with that, any questions? We have time. And then I'm giving my email in case you have questions that you want to ask that don't get answered today. I'll also help answer towards the end or whatever. So like plenty of time for questions.
Thank you, Zane. I have a lot of questions. And I would say maybe take down your screen sharing so we can see each other. My first question is, and shout out to our friends at Save the Redwoods League. I know they've been really instrumental in helping support this kind of research. So I want to make sure we call them out. in addition to the universities that have been supportive of this. So, stepping back, I'm curious, why did you and Save the Redwoods League feel like it was so essential to map the genome of the redwoods? A, and B, all this cool stuff that you just told us, I kept thinking throughout your remarks, how unique is this? Or is some of this very typical within the plant world? It's just very different than what we might have learned in our genetics class, back in college or whatever, from a human standpoint.
Sure. So to answer the first question, the league wanted to do the Redwood Genome Project as a way to ideally where we can go out and sequence a bunch of trees in the forest and know something about, based on their sequences, something causal about them, right? Like
Mm-hmm.
maybe their genetics make them more predisposed to drought, or maybe it makes them
Yeah,
more resilient to drought, right?
yeah.
Or any of those things. That work is still occurring. The hexaploidy problem and this diploidization problem becomes really intense with all these different copies. And that has made it really difficult for people to work on. That was not my part of the project. I did a little bit of work on that. And we published, there's been some work published on that, but not a lot. And the Redwood genome is 9 times the size of the human genome. It's way more complex. We have 6 copies of everything, and those 6 copies are neofunctionalizing and diploidizing, and so becoming more and more different in the different populations. And so it's just a mess. So I think there's going to be more work done on this.
Mm-hmm.
There are some more publications that are coming out over the next couple of years, including this clone publication that hasn't been published yet. We're still working on a few different aspects. To answer your second question, there have been maybe a handful, like probably fewer than 10 studies that look at different tree species and mutations that occur in them. And depending on the method, there's a lot of complexity that I'm skipping over, that depending on your method, you may find fewer mutations than are actually relevant at a reproductive level, like I'm trying to talk about here. But we know that taller trees seem to mutate more often, and older trees seem to mutate more often, or they can accumulate more over their age. And so we think the age has something to do with it. It might also have something to do with cell divisions. Um, but I would say, yeah, it's understudied for sure. And so
So
if we
within the tree world, redwoods may not be that unusual, or they might be on the more extreme end, or do we know?
they might be pretty unusual. So this is redwoods have the highest mutation rate of any eukaryotic organism measured thus
Mm,
far. And so eukaryotes are any multicellular organism, basically. Redwoods have the highest
wow.
mutation rate per year and per generation. And so that's pretty weird if you ask me. And then also because they're hexaploidy, because they're hexaploid, they are kind of more relaxed to mutations. And so that could also factor in. Um, we just don't know enough. What I would encourage for people in general, if people are wanting to look at forestry genetics and forestry genomics, is to not just sequence individual branches from individual trees, but to at least have 2 branches from each tree to get some idea of the genetic diversity that's within a single tree, especially if it's long lived.
Yeah. So, we go to often with many of our webinar speakers, what does it mean for land managers like us or entities that want to be advocates with other public land managers? And I'm curious, would you expect to see mutations occurring in response to climate change, or climate change selecting out certain genetic traits? Is it too early for that?
So maybe. So if branches are reservoirs of genetic diversity, what does it mean when we have something like the CZU fire? What does
Right.
that mean to genetic diversity? Or what does it mean when we have logging? What does that mean to genetic diversity? If we're describing trees as big populations of a bunch of branches that could have unique genetic combinations that could end up in a seedling, keeping as many of those branches around kind of matters for the genetic resilience of the species.
Right.
So, from my perspective, uh, I think having as many branches as possible is the best possible thing. And so when you have fires like this in Big Basin, trying to reduce that through something like controlled burns matters a lot. I don't know about thinning, because thinning obviously is getting rid of branches.
Yeah.
And so, like, I kind of think leaving that up to nature is maybe a better approach so that selection can act on what trunks are better or worse, what branches are better or worse, what mutations might exist that are better or worse for survival in the 21st century and beyond.
Hmm. So from a land management standpoint, and we've been big advocates of reintroduction or introduction of prescribed
Mhm.
burning or
Mhm.
utilization of prescribed burning, I should say, but also increasingly of the need for mechanical thinning, i.e., logging, just to deal with the unnatural density in the forest that exists right now and to avoid
Mhm.
the catastrophes of the next
Yeah.
CZU. I'm hearing you say that maybe we need to be really mindful about in that process that we are eliminating genetic diversity that may hold, the branch that holds the reservoir of genetic material that's going to have the redwoods that can survive in hotter and drier climates.
Mhm. Yeah. Basically, the branches make either a really large population size if there's a lot of them, or a very small population size if there's not. And so populations, the ones that are in trouble from climate change are the ones that are small,
Yeah, because
right?
they're
The ones that are
hardy.
vulnerable. And so I think having a big population size matters. Um, I so I think controlled burning might be the better way to do it, because
Yeah.
that is still, I guess, to a certain degree human selection, but it's also natural select. It's sort of like, like, let's see what branches survive our treatments. I don't know. It's really challenging. Then another
Yeah.
thing I think about is, like, redwoods have been around 180 million years. They've been doing their sprouting thing for 150 million years. I was in Dinosaur National Monument uncovering a fossil burl,
Oh
that
my
was kind of
gosh.
cool. And, like, they've been, like, I don't know. And they've been in California for 5 million years or thereabouts. And humans have only been here about 15,000 years. And so I tend to think, like, trust the redwood tree. It knows what it's doing. It's been around for a while.
Well, and I guess
But
you're
that's more of a
bringing
hands-off approach, and I know that that's, I don't know, controversial, or that's just my opinion.
it's hard to know what the right thing to do is given the
Mhm.
circumstances and given how
Mhm.
disrupted the natural forest ecosystem is, both because of historic land management practices and because of what's happening to the
Yeah.
climate. And so we struggle. It's why I ask almost every
Yeah.
member guest at our webinar series, tell us what we should be thinking about given the lens that you bring to it. So it's
Yeah.
very appropriate to be raising it. It's just question mark.
Yeah.
Um,
One thing I'll say is, like, suppression is a land management decision as well,
mhm,
right? And, like,
mhm.
and we kind of have to, because people live, like, the CZU fire threatened a lot of homes. It took a lot of
Right,
homes, right?
right, right.
And so that's a tricky thing,
Yeah,
because by us suppressing or saying, oh, no, no, we don't want the forest to
yeah,
burn, that's
yeah,
that's also a decision. So, like, we make a
yeah,
decision either way.
yeah,
It's,
yeah, for
it's
sure.
Schrodinger's cat, dead or alive, we're making decisions, right?
Right,
So,
right. I'm curious, it feels to me listening to this incredible diversity and rate of mutation, it leaves me actually with great hope that the species is going to have enough diversity within its population that as it faces these changing conditions, there will be some version of redwood branch somewhere that can withstand some of these things. And it may end up looking very different or have some slightly different features or not so slightly different features. Do you think that that sort of optimism that I'm expressing is warranted?
yes. Yeah, I think it's very optimistic for the redwood species. I worry about other species, or like I think focusing on other species in the redwood forest might matter more than focusing on redwoods. So things like Douglas fir, CZU complex took out like 98% of them.
Yeah, I
Big
was
Basin
thinking
is
about that when you were talking
virtually
about
with
that giant Doug fir.
yeah, it's virtually without Douglas fir. And there are
Yeah,
very few seedlings there of Douglas fir seedlings. So like bringing Douglas fir back matters. I think marbled murrelets in Big Basin, that's the southernmost end of their range, and they're found nesting in more Doug fir branches than redwood branches down here. And so by the Doug firs being gone, like we need them back. They grow
yeah.
faster than redwoods.
Well, and we've had conversations with some forest ecologists who will say just pulling out a species like that, eliminating all the Doug fir, don't think that it's not going to have an impact. It is. It's not like they just got plucked out and there's no implication for the ecosystem. Um,
Mhm.
well, perhaps a reminder to approach all of this with some humiliation or humility, I should
Humility. Yeah, yeah, yeah. Yeah, yeah.
humiliation if we fail to bring humility.
Well,
Um,
then we would call it a strategy, wouldn't we?
yeah.
So we need to
Um,
be careful that we're using good strategies, not strategies.
yeah. Yeah. Right. Right. So I want to give, I know we're running out of time, but I want to give the audience a chance if there are any questions from the Q&A section that we want to share with Zane. And I want to assure the audience that we will answer some of these questions offline.
Yeah, Zane, thank you. We have lots of questions and really
Mhm.
appreciate your willingness to follow up with answers after this webinar.
Yeah, yeah.
The one I wanted to focus on is really about a little bit about terminology and how we think about the human relationship to trees. We talk at Semper Byron's Fund a lot about second growth trees. This idea that when trees are cut down, there's trees that emerge from that landscape. Um, how does that fit the genome mapping model that you're looking at in terms of, are these really second growth? Are they actually clones of the nearby stumps of trees that were logged? Is there another way that we should be thinking about that transition?
So when I say clone, I mean a developmental clone. It all developed from one seedling. But what happens is once that embryo grows, even if you looked at different cells of the trunk on different parts of the tree or different sprouts on different sides of the tree, they will have different mutations in them. And so they're mostly identical. They're 99 point whatever percent identical. But there are quite a few mutations that occur in any side of the tree. And so, you know, um, they are going to be different. And then as they grow, they're going to accumulate mutations as well. So I don't know. Like you can keep splitting hairs and say like, oh yeah, the cell's the individual. Oh, the DNA strand is the individual. The chromosome's the individual. Um, yeah. So there comes a point where it's like, do we really need to be protecting branches? Like, okay. So what I would say is I still am a firm believer in let nature do its thing, let selection happen on the environmental landscape without us sort of coming in and selecting for it. But, we make those decisions.
Are you thinking, and is there a movement afoot? I'm going to ask one last question and then we'll have to wrap up.
Mhm.
Is there a movement to try to find trees, for example, that we think are more drought resilient and purposely plant them, say, in drier parts of the range? I know, I imagine that's part of what the league had in mind when they first initiated this study.
Mhm.
Hearing from you at the level of complexity, I'm starting to have questions about whether that's really a viable option. And so I'm just curious how much practical application you think there is around trying to preemptively plant trees, diversify the population in order to mitigate the impacts of climate change. Are we anywhere near being able to do that?
Nothing that we could control or that we would fully understand the implications of.
Yeah.
Um, I don't think it would be that bad of an issue of planting some trees that don't exist in this particular location from somewhere else. But I do think it's important to have some level of well, they're hexaploid, they have a lot of diversity. I would, that's a hard question. I mean, I would use cones and seeds and things. I wouldn't worry about sequencing things and then planting based on a sequence.
Yeah.
I would worry more about like using cones that exist already.
In a drier, coming from a drier area, perhaps, or something like that.
Maybe. I do think you could have some issues with inbreeding in the southern end of the range, and in like Oregon, there could be inbreeding issues where if you were to transplant one's inbred individuals from both ends and kind of transplant them like this, that hybridization would then allow for more alleles more combinations of alleles to be selected for like what works best.
Yeah.
So maybe.
Yeah. Okay. Well, we're going to have to bring you back.
Sure.
I have so many questions, but thank you. I want to thank you, Zane, Dr. Moore, for joining us. Thank you for your work in the film Giants Rising. It was really fun to see you there as well. And we will definitely bring you back. And to our audience, thank you for joining us. Please, again, look for us on June 24th to have a discussion with some of our with our colleague, Rachel Dan, who will talk about some of the policy issues that are relevant to what's happening on the ground and in the climate and how decisions being made in Washington and Sacramento may impact that, and how you as an audience member can participate. So thank you all. We'll see you again under the Redwoods. And thank you again to Sharp Investments for sponsoring this webinar series. All right. Thanks.